Hello, everyone, and welcome to another very exciting seminar in our series. My name is Julia Darolti. I'm a postdoc in Judith Mang's lab at the University of British Columbia. And before I introduce today's speaker, I just want to remind everyone that, as always, there will be a Q&A session after the talk. If you would like to post questions for the speaker, you can do so in our dedicated Slack channel, where you can also upvote questions that you would like to see answered. Today, we are very fortunate to have with us Scott Edwards, Professor of Organismic and Evolutionary Biology and Curator of Ornithology at the Museum of Comparative Zoology at Harvard University. Scott's research focuses on diverse aspects of evolutionary biology of birds and of non-avian reptiles, including behavioral evolution, comparative genomics, systematics, as well as disease ecology. Scott has made key contributions to the understanding of the major histocompatibility complex in birds and how this plays a role in adaptive immune response and in mate choice. He and his lab have developed novel phylogenetic theory and methods and have conducted extensive comparative work on birds and other reptiles to uncover the evolution of feathers and the flightlessness. Scott has received numerous awards and distinctions for his work, including being elected a Fellow of the American Association for the Advancement of Science, member of the U.S. National Academy of Sciences, as well as of the American Philosophical Society. And today he is going to tell us more about wings, feathers, and flight, and how phylogenetic approaches can help identify the genetic basis for phenotypic variation across species. So thank you very much, Scott, for accepting our invitation, and over to you.
Thank you, Ilya. Appreciate it. It's great to be here. It's a little strange not seeing anyone out there, but I'll assume there's at least someone listening to me. Thanks again. I'm really honored to participate in this series, which has really made the summer sort of tolerable, I think, for a lot of folks. Yeah, without further ado, I'll go ahead and share my screen and introduce the topic for today. As Yulia mentioned, I am an ornithologist at heart, and so most of the work we do in my lab focuses on bird evolution. And today I want to tell you how we're trying to use comparative genomics to understand bird biology, diverse aspects of bird biology. And this is sort of precipitated by a recent review paper, opinion piece that I wrote along with Stacy Smith and Matt Pennell, who is also at UBC, and Casey Dunn, really trying to push this idea that we can actually use phylogenetics to understand the genetic basis of traits. At recent meetings that I've been to, including the evolution meetings, you see lots and lots of work going on, people using phylogenies to look at ecological traits or biogeography, but there's actually relatively little work going on using phylogenies to understand the genotype to phenotype map. And that's what Stacy and I and others got excited about. And hopefully our opinion piece will spur some more activity in this area. And so what I hope to show you today are some examples from our work about this PhyloG2P approach to make links between genotype and phenotype for species that really can't be bred in the lab. So for example, we can contrast the traditional approach to mapping genes here in the center of the slide with this new sort of forward genomics approach, in which instead of creating a population that's segregating for the trait, as we would with a model species, instead we find a clade of interest that is sort of segregating for the trait, where you might have a unique evolution of a trait, or you might have convergent evolution, or multiple appearances of a trait. And we can use comparative genomics to sort of look at what parts of the genome are correlating with that trait. And I'm sure you can imagine a host of difficulties with this approach, but what I've been impressed by is just how powerful it can be. For example, one example that we highlight in our opinion piece was this work by Gil Bejarano from Stanford and his group, in which they put together a series of phenotypic traits. In fact, they looked at about 3,400 phenotypic traits. And actually, I should change this word here from human to mammalian. They actually had, from a large data set, 3,400 mammalian phenotypes. And they looked for the coevolution of those with about 260,000 regulatory elements across the genome. So this is a nice example of sort of big data. You can see on the left here, we're starting the whole process from identifying these conserved non-coding elements, which I'll talk about later. That's what CNE stands for. And you can see on the left, we're finding patterns of loss or conservation of these elements across the tree. And then we're matching those patterns of loss with phenotypic changes or loss of a phenotype, okay? In this case, they found an interesting match between a set of regulatory elements and the pedal phalanges of the foot across different mammals. Then on the right, you can take this element, find out what gene is nearby, and that's the gene that it likely regulates, although it could potentially regulate other genes. And you find what that gene does. And remarkably, in this example, you actually see a correspondence between the phenotype changing and the nearby gene. And this corroborates the approach. And in fact, they found some extraordinary associations between skeletal elements, elements of the middle ear, really, really striking associations that make you feel that this approach is actually working. It's not just noise. And so what I hope to do today is to use this approach to contrast the sort of two hypotheses for the evolution of traits, which were introduced to the literature by this seminal paper back in 1975 by Alan Wilson and Mary Claire King. And they suggested, based on their comparisons of humans and chimpanzees, that the genetic differences between these species were far too slight for genes to explain the phenotypic differences. I mean, at the time they were looking, they could look at electrophoretic variants of coding regions of the genome. They could look at whether chimpanzees and humans shared certain alleles, or they could even look at some protein sequences. And what they found is a conclusion that we're still with today, namely that humans and chimpanzees differ by about 1 or 2% at the genomic, at the sequence level, very slight differences, which basically led them to the conclusion that it must be the way in which genes are deployed, namely gene regulation, which distinguishes these species rather than the sequence of genes themselves. So let's see how this plays out in a couple examples from birds. First example involves some work by my former graduate student, Baldwin, who's in Germany now. And Maud was interested in the evolution of taste reception in birds, and she still has a very active program in Germany looking at this question. And you can see here that we've got two taste receptors, umami on the left and sweet taste on the right, which are both underlain by small multi-gene families consisting of these heterodimers, these so-called T1R, T2R receptors. Now, in nature, we have some really interesting natural experiments where certain species have actually lost one of the other receptors. So, for example, pandas, whose diet is exclusively bamboo, have lost their umami receptor. They can't taste meat or amino acids because they don't have this receptor. Similarly, cats, which also evolved, domestic cats, which also evolved from a carnivorous ancestor, have lost their sweet taste receptor, despite their preference for things like milk. They can't taste sugars. And so these experiments suggest that a lot of really interesting questions about the genotype-phenotype map. Now, birds, it turns out, we know evolved from a highly carnivorous lineage of dinosaurs called the theropod dinosaurs. And what's remarkable is that in no bird species can we find a sweet taste receptor. And this perhaps makes sense because birds, their lineage goes back to carnivores such as T-Rex and Archaeopteryx and other theropods. Even hummingbirds, which we know to be able to discriminate sugar water from plain water very, very efficiently, do not have a sweet taste receptor in their genome. So Maud wanted to ask, how is it that hummingbirds can taste sweet if they don't have a sweet taste receptor? And she basically compared the genomes of hummingbirds and swifts, the closest living relatives of hummingbirds, as well as chickens. And of course, she also corroborated the hummingbird's ability to taste sugar through field experiments, and confirmed that whereas they could taste sugar, swifts and chickens could not. And to cut a very long and laborious story short, Maud was able to show that the umami receptor in hummingbirds had undergone a series of amino acid changes which conferred on it the ability to bind and transmit signals from carbohydrates, including sugars. So here we've taken a receptor who's adapted for detecting meat and amino acids, and through a series of point substitutions have modified it to be able to bind carbohydrates. So it's a great example of what Stephen Jay Gould called an exaptation. A trait formerly adapted to one function gets exapted to perform another function. Excuse me. And I would say is a point I would say in favor of the importance of genes for phenotypic traits. So it somewhat runs against Alan Wilson's conjecture that gene regulation is more important. And so we can use, this is an interesting example where we've compared a small group of species to strongly implicate a particular gene in the origin of a phenotypic trait. And of course, sweet taste has probably enabled hummingbirds to diversify into the over 300 species that they comprise today. Now, of course, genes are only a small part of bird genomes and other genomes. And we know, for example, that genes comprise only about 2% of the genetic material. And so it behooves us to understand what's going on with the other 98% of our genomes, this so-called dark matter of the genome. And some have postulated that this dark matter is a vast regulatory network which might be focused on understanding the might be focused on orchestrating the deployment of genes and when they're expressed. And so one sort of foothold into this complexity is to look at a class of loci called conserved non-exonic elements. These are essentially the same as the conserved non-coding elements, which I mentioned in the earlier slide. And we can identify those through comparative genomics. We can look for regions of the genome that are conserved between species, such as you see here in black in this diagram from the genome browser, and yet do not occur within a coding region. These are good candidates for being conserved non-exonic elements. And we know that many of such elements could, in fact, comprise what we call enhancers. Enhancers being a class of non-coding elements that recruit things like transcription factors and other aspects of the transcription machinery to express to drive the expression of nearby genes. And so these conserved non-exotic elements are really exciting as sort of a first look at what this regulatory landscape of the genome might look like. And so basically, and for those of you who are interested in how we might identify these, we use a so-called hidden Markov model to basically scan the genomes of a series of species and look for regions that are evolving slower than a putatively neutral rate. And so in our case, we aligned a bunch of genomes from birds and many reptiles and even frogs and fish. And we can pull out regions that are highly conserved between these species by looking for signatures in the genome that produce a tree whose branches are demonstrably shorter than the so-called neutral tree. And the neutral tree we can get from any subset of the genome that we think is putatively neutral. It could be silent sites, or it could be silent sites of genes, or it could be transposable elements that are dead, or who knows what. Anyway, we use this approach to try to understand the role of gene regulation in the origin of feathers. And feathers, of course, are a hallmark of bird biology. Of course, we now know that feathers arose deep in the theropod lineage and don't necessarily distinguish modern birds from their dinosaur ancestors. But these days, scientists can tell you not only about the fine structure of feathers in the fossil record, but even about their colors by looking at patterns and sizes of shapes of these organelles called melanosomes. And so what we did was to look at the pattern of origin of genes associated with feather development on the one hand, and conserved non-exotic elements that are associated with those so-called feather genes. So we found we went through the literature, much like Gil Bejarano's group did with the mammals, and we found almost 200 genes that people had implicated in feather development. And in the genomes, we were able to detect over 13,000 conserved elements that were close by these 200 feather genes. And what you see below is a screenshot showing an example of one of these feather genes called S-Hox. And you can see how the exons of S-Hox are conserved all throughout vertebrate evolution, going all the way from birds to mammals to fish and frogs. Those are what these black lines mean. And intriguingly, there are elements, these short conserved elements outside of the gene, which are variably retained across either birds in red, reptiles in green, such as this one here, or amniotes in brown, such as these here, or perhaps even all vertebrates, such as this CNE here, which is found in birds, but also in fish, which suggests that it was present in the common ancestor of those lineages. And we basically asked which of these parts of the genome is undergoing a burst of evolution coincident with the origin of feathers. That was our question. That's how we designed our study. And this is what we found. First of all, on this graph, you see a sort of a timeline where you have vertebrates on the left and chickens and ducks on the right, modern birds. And on the y-axis, you have sort of a measure of relative origination rate. How many new elements do we see in these various parts of the tree? We know that feathers evolved about 220 million years ago. And for example, the genes themselves, as you saw in that S-Hox example, many of the genes associated with feather development are actually quite old and were in the lineage of birds long before feathers evolved. And so we can suggest that actually most so-called feather genes actually predate feathers. There is one group of genes, the beta-carotene genes, which did undergo a burst of gene duplication coincident with feather evolution, and that we shouldn't downplay the importance of those genes. But on average, the average feather gene is much, much older than feathers themselves are. Then we can look at these conserved elements, and remarkably, we find a burst of evolution coincident, one with this time frame right here in the middle of the slide, which is perhaps coincident with the transition from water to land in amniotes as amphibians evolved into amniotes. And perhaps more intriguingly, another burst that's coincident almost perfectly with the origin of feathers themselves. These are all these bird-specific elements that you saw on the previous slide. And so we can suggest, in fact, that the feather gene enhancers may have evolved roughly at the same time as feathers themselves, which might spur us to conclude that gene regulation, as opposed to genes themselves, are perhaps more important in the evolution of feathers than changes in the coding sequences of genes. Again, we shouldn't downplay the beta carotenes there, but we were the first to suggest that actually it's how genes are put together, not evolution of the genes themselves, that might really have been underlying the evolution of feathers. We also looked at what parts of the genome underwent a burst of evolution, what parts of the regulatory toolkit underwent a burst of evolution on the branch leading to modern birds. And that's what's indicated by this star. And what's interesting is that we find variation in the density of these conserved elements that appear on the avian branch. That's what you see in this plot below here. And these red regions are regions that are where the birds have higher than background rate of origination of these conserved elements. And what's cool is we found one region of chromosome 7 in chickens that had an unusual spike in the density of these conserved elements. You can see there on the right. And it turns out that that high density of conserved elements is very close to genes that are involved with things like limb size evolution or body size evolution. These insulin growth factor binding proteins are well known in the agricultural literature to be associated with body size evolution of domesticated species. And so we suggest that these changes in regulatory elements influence the deployment of things like insulin growth factor binding proteins, or GLI2, a gene associated with limb element development. And perhaps we're involved with the transition of theropod dinosaurs to modern birds. Okay, so we've seen two examples, one involving taste receptors and one involving feathers, where we can use comparative genomics to implicate certain genes or certain parts of the genome with a phenotypic trait. Let's now move to our last example, where I'll talk in detail about the loss of flight in birds and how we can use the PhyloG2P approach to understand the genomic drivers of loss of flight. And this focuses on a group known as the paleognaths. I'm sure many of you have seen a paleognath. You might not have known it, but if you've ever seen an ostrich in a zoo or even out in the wild, or emus or kiwis, you are looking at paleognaths. And now some of you may even have seen this intriguing species in the middle, which is also a paleognath, but it turns out it can fly. It's a member of a group of birds called tinamous. And tinamous are volant species. They can fly, but their skeletal characteristics clearly link them with these flightless so-called ratites. The ratites are so named because their keel is missing, their sternum is missing a keel. So for example, you see here these sterna of an emu and an ostrich, they're missing the big keel that we typically see, typically see in a flying species. Of course, ratites also differ in a lot of other ways: body size, differential loss of limbs. So for example, the moas of New Zealand, they're actually the only ratite which that's lost its forelimbs entirely. There's absolutely no humerus, radius, or digits in moa skeletons, whereas things like emus and ostriches actually retain some of these elements. And so we wanted to use this approach to look at what parts of the genome may be co-varying with loss of flight. And we were able to do this because we strongly suspected that flight had been lost convergently in the ratites. And this was based on earlier work done by others, which surprisingly showed that tinamous, rather than serving as an outgroup to the entire ratite clade, as was traditionally assumed, tinamous actually fell inside the ratite radiation. Big surprise. Ornithologists were understandably perturbed because this seemed like a radical departure, but this was really, actually, Mitchell et al. Did some of this work, but even earlier, back to 2008, folks like John Harshman and other folks working on big bird data sets were able to discover this. And this raised the possibility that flight could have been lost as many as 6 different times, which is a great, very fertile ground for applying the PhyloG2P approach. And so what we did was to, this just shows that we looked at, it's possible to imagine a scenario of character change in which it's more likely that flight was lost multiple times rather than being lost in the ancestor of all of these birds and regained in tinamous. And so this plot here just shows the parameters of character change, rate of loss and rate of gain, that actually favor multiple losses over a ancestral loss and then a gain in tinamous. Of course, just thinking biologically, many ornithologists are more comfortable with multiple losses of flight than a regain of flight because it's such a complex character. So we went and did a bunch of sequencing of a bunch of different paleognaths. They actually have somewhat bigger genomes than flying birds for interesting reasons connected to their ecology and population biology. We sort of inherited a genome from a little bush moa which was created by Alan Baker, our collaborator, who sadly passed away at the beginning of our project. But he had produced a fairly good draft genome of a little bush moa from New Zealand. We put all these genomes in a big browser, and you can actually peruse this browser if you want from your desktop using links that we provide in the publication. But you can see that this is a nice way to view different genes and how the alignments are working. And we used a really nice tool developed at UC Santa Cruz called Progressive Cactus, which basically is a phylogenetically informed alignment tool, which allows you to look at all parts of the genome of every species rather than just those parts that are present in the reference genome. Most people have a chicken as a reference genome, and then they'll sort of map everything to that one genome. But that makes it difficult to look at parts of the genome which are unique to non-chicken lineages. So this progressive cactus is a really nice tool that has been, it's gonna, I see a lot more wider use, I suspect. We first wanted to build a tree, and so we had confirmed some of these earlier results. So we got a huge number of markers from all over the place, ultra conserved elements, introns, conserved elements, all with different evolutionary properties. And we came up with a set of hypotheses that really made us realize that it was really the rheas here from South America, which were really the remaining enigmatic and conflicting lineage in previous studies. You can see it bounces around the tree with variable support. And so when we put all our data together, and I think importantly, analyzed it in a coalescent framework, most previous studies had simply concatenated genes together. We tried to model the biological process and allow different markers to have different trees. We came up with a very consistent tree that was very robust and very robust to perturbations in the data set. And this tree actually had rheas branching off just before the emu cassowary and kiwi clade. Very short branches here, at least in this diagram, but robust branches nonetheless. I mean, there's a lot of information in these short branches, given the number of markers we were looking at. And you can see that there's, depending on your predilections, you could imagine 3 losses, you could imagine 5 losses, you could imagine different numbers of losses, depending on how you interpret the distribution of these species and where they might have flown or where they might not have. And so a lot of the conclusions that I'll mention to you are pretty robust to what we assume about the number of losses. And here, of course, is the loss and gain scenario, which we think is unlikely. Just to give you an idea of how many different signals we saw in this data set, we found, especially around the rhea emu kiwi clade, we found a really almost even distribution of markers favoring one of these 3 topologies. And so you can see that there's a slight preponderance of markers in green here that favor this major topology, but that the number favoring these other topologies is almost as high. And this is precisely what you would expect if this process known as incomplete lineage sorting is operating. And this, of course, refers to the idea that processes of drift in ancestral lineages don't proceed fast enough for there to be a clean sorting of lineages between speciation events. And so these coalescent methods have been developed to accommodate the expected heterogeneity in gene lineages in rapid radiations like this. I'll belabor you with one more set of tree statistics, just to impress upon you how having a large number of markers and markers that are analyzed in a coalescent framework is really, really helpful. You can see here that as we add more markers, the emu cassowary kiwi clade gradually increases in significance in terms of bootstrap support, whereas other hypotheses fade away. And when we concatenate genes, such as you see down here, we see a real uneven mix of signals. It's really hard for concatenation to deal with these heterogeneous signals. And in fact, you can see that the one signal that does appear to rise up is a different one from what we expect from a coalescent framework. So the bottom line is we think we're right, concatenation is wrong, only because it doesn't really model the evolutionary process of these markers very well. All right, let's, I'm not going to talk about this. This is, let's get to back to the biology here and remind ourselves that we're interested in evaluating the relative role of genes or gene regulation in the origin of this trait. And I won't share with you a lot of the tests that we did with protein coding regions, except to say that we didn't find any really convincing signals of protein coding changes that were associated with the convergent loss of flight in this group. We didn't find any evidence for pseudogenes, which was surprising because in some traits like loss of teeth in birds, there's clear evidence for pseudogenization of genes important for building teeth. We didn't find any evidence at all for actually convergent evolution of amino acids in the lineages leading to flightless species. We found some evidence for convergent adaptive evolution or convergent acceleration of genes in lineages that have lost flight. And those shouldn't be discounted. I think there's, we found about 200 genes that putatively could be associated with the convergent loss of flight. But remarkably, they didn't coalesce into a clear statement of the function of those genes. And so there was, when we did the gene ontology analysis, or looked for what functions are enriched in this set, we found very little of that made any sense. Nothing involving development or limb development, or so it was not a very strong case. So we looked again towards the regulatory landscape, and here we undertook a series of studies. Again, here are CNEs. I won't tell you about that again. We looked at a total of almost 300,000 of these CNEs, and we constrained them to be a bit longer than 50 base pairs, just so we could look for things like rate shifts in different lineages. And we basically looked for regions of the genome that were highly conserved in flying species, such as tinamous and the neognaths here. Those are modern birds besides the paleognaths. And yet were variable or accelerated in the ratites specifically, the flightless group. We interpret acceleration here either as natural selection accelerating the element, or simply a loss of constraint and a return to a neutral rate of evolution. And you can see here that there's relatively few substitutions among this conserved group. Basically, similar sequence in this 250 base pair region over 100 million years or so of evolution. But yet there's a sort of a detectable uptick among the flightless birds. So it was elements like this that we went around the genome trying to detect. And without going into a lot of detail, we worked with a statistician named Jun Liu and his students at Harvard to develop a very robust Bayesian model that was tailored to finding convergent rate accelerations. Again, I don't want to go into the details too much. It's a really exciting approach that allows transitions between different states, for example, conserved state or a background state or an accelerated state. And it can allow the easy testing of hypotheses as to whether the rate increases are occurring in the specific target lineages, such as species 1, 2, 3, and 4, or erroneously in this entire clade consisting of both target and non-target species. We use a series of Bayes factors to do that, and we showed that this approach is actually more sensitive than tools like BEAST or other methods to detect rate shifts. It's very good at detecting convergent rate shifts, where other methods would tend to call this entire clade accelerated. Here's an example of another of an element that's undergoing a accelerated rate shift in MOAs and RIAs, for example. You can see the branches here that are the state that each branch is in here. This says there's been adaptive evolution or relaxation of selection. Here's some more examples. I just love looking at these pictures. It's remarkable to me that you can find regions of the genome that are accelerating specifically in these very few lineages of ratites. Here you see our estimates of the rates and the Bayes factors associated with the preferred model. Anyway, what we found was that many of the genes that were near to these so-called ratite accelerated regions were highly enriched for developmental processes that might be implicated in limb loss. So for example, things like TBX5 had been studied already by developmental biologists. You can see a few Hox genes here, other kinds of genes associated with skeletal developments. And so this suggested that we were getting some interesting signal with these conserved regulatory elements, that they might be associated with developmental processes. And in fact, we confirmed many of these genes. We confirmed their expression in fore and hind limbs of chickens in this case. These are genes whose regulatory regions nearby are undergoing acceleration. So that was suggested that some of these genes could be involved with some of the limb adaptations associated with loss of flight. We also looked at regions of open chromatin using a method called attack-seq. And what's important here is that you can imagine how any given element or gene, we might not know what part of the body or what part of the developmental process it works on or is involved in. Attack-seq is a really nice way to implicate specific regions of the developing embryo where a particular regulatory element might have activity. And so I don't know if you're familiar with attack-seq, but basically it identifies regions of the genome that are in the so-called open chromatin, namely they're not wound up in nucleosomes, and therefore can be accessible to things like transcription factors and other aspects of the transcription machinery. And so we looked specifically at fore and hind limbs of developing chickens and many other tissues to see what the chromatin landscape was across the genome. And here what you see is that across about 8 different tissues, including forelimb and hind limb, flight muscles, sternum, we see an overrepresentation of these conserved elements in open chromatin, okay? So in this slide, we're only looking at about 1.5% of the genome that's in open chromatin. And yet we find there's a disproportionate representation of these conserved elements in that 1.5%. So this says that these are probably active enhancers because we're finding them in open chromatin that's accessible to transcription factors. And so the kinds of things we were looking for are regions of the chicken genome that conserved elements that were in open chromatin. That's what these black areas indicate. Here's a conserved element here that is actually accelerated in the ratites. And remarkably, not only is it accelerated, but RIAs, for example, don't show open chromatin in that area. And so this says that that element not only has undergone an increase in evolutionary rate, but its chromatin landscape has changed as well. And this further implicates it being associated with, in our case, forelimb evolution, because the attack-seq landscape will differ from organ to organ. It's a remarkably dynamic process. And we ended up basically narrowing down our list of candidates by looking at the union, the intersection of rate accelerated elements and ataxic, and also other epigenetic marks from other work. And we were thereby able to take a very complex, large set of elements and narrow it down to a smaller number that really could be interrogated in much more detail. And so what we did was to take a few of these high significance CNEs and inject them into developing chicken forelimbs, basically to ask if they could drive gene expression. And so what you see on this top row is an example of the chicken enhancer. All these are done in chicken embryos, but you see the chicken version of this enhancer. We see that the injection is successful because it's in red. This is work done by my graduate student, former graduate student, Phil Grayson. And that it's actually driving gene expression as indicated by this green fluorescence here. Here, now we'll put the Tinamou version of that enhancer in the chicken embryo. Also a positive control here, and it also drives gene expression. However, when we put the RIA version of that enhancer, and remember that's an accelerated region, despite the positive control, it does not drive gene expression, which strongly suggests that it is differentially driving gene expression in volant and flightless species, perhaps due to the sequence evolution it's undergone. So we've taken this PhyloG2P approach and gone all the way from identifying a subset of regions of the genome that look interesting phylogenetically and then functionally testing them through this differential enhancer assay. And I won't, I'll just simply say now that our ongoing work that Phil Grayson is finishing up now is to look at chromatin and transcriptome expression across the suite of 5 species. Despite the beauty is that, you know, people actually eat emus and eat rheas and ostriches, so you can actually get eggs and embryos of these things pretty easily. So it's a really nice set of species to look at evolution of gene expression as well as evolution of the chromatin landscape, and that's what we're writing up right now. And so I'll just quickly go to the conclusions here. And on this last part, we've seen how we can use the phylogenetic tree to pinpoint regions of the genome that look interesting. And importantly, I think we have to accommodate some of the details of the biology of the markers we're using to build that tree correctly. And we've shown how we can take some of these non-coding elements and show that they co-vary along with the phenotype of interest. And then we can finally use functional approaches like ataxic to really nail down their possible role in limb evolution. And so I would just like to thank this amazing team that we worked with. You can see Tim Sackton, who really led this work, and Phil Grayson did a lot of the bench work. Alison Cloutier, as well as Michelle Clamp, did a lot of the computational work along with Tim. And we worked with Cliff Tabin. We're continuing to work with Cliff Tabin on the developmental aspects. And Julia Clark, a paleontologist, kind of kept us honest in terms of our morphology. And I was pretty much the happy bird watcher behind all of this cool science going on. So thank you very much, and I'd love to take your questions.
Great, thank you so much. Amazing work. While we wait for some more questions from the audience, I had one of my own. I was wondering if in your comparisons across species, have you looked at all at whether there's a link between genome size and flight, whether because of variations in metabolic costs, maybe there's associated with powered flight, maybe there's some differences in genome size across species?
Right, I alluded to that a little bit. Yes, flightless birds tend to have slightly larger genomes than volant species, and we think it's probably a sort of a demographic effect. It could have to do with metabolic rates, because we know that many of those species do have lower metabolic rates. We were thinking that, Michael Lynch has this whole framework for thinking about non-adaptive mutations and in populations that are relatively small. And it may be that because ratites are large-bodied and flightless, that their effective population size might be smaller than those of in general of flying species, which would result in a increase, a bit of a sort of a thwarting of natural selection, and an increase in the accumulation of slightly deleterious elements like transposable elements or other insertions. So, but I would be really interesting to look at in more detail the precise causes of this modest increase in genome size.
Yeah, that'd be very interesting. And sort of related to that, someone is asking whether there's any role of transposable elements or horizontal gene transfers in feather evolution.
Well, that's a great question, since we know that some regulatory elements are derived from transposable elements for sure. And we didn't really look at that. It would be quite interesting to see what fraction of our 13,000 feather-associated regulatory elements might be derived from transposable elements. That actually is a really great idea, and we hadn't thought about doing that yet.
Cool. There's another question saying that the chicken genome was recently re-annotated, revealing a large number of genes on small chromosomes that had been previously missed. Does the rediscovery of these coding regions have an impact on your results? Are there existing regulatory features on those small GC-rich chromosomes?
That's a great question. I mean, and I think all of this work was done with either chicken version 3 or 4. So yeah, it was probably a bit incomplete. And it would be quite interesting to revisit it. Send me an email, we'll talk.
Great. Another question is, do you think that the patterns you observed in the ratites in terms of cis-regulatory elements could also be observed in other more distantly related flightless birds, such as penguins?
My guess is that they could be, and I think that would be a very interesting project. Yeah, absolutely. It'd be interesting to look for elements that are accelerated both in the ratites and in things like penguins. There was, you might remember, a paper on loss of flight in Galapagos cormorants a few years ago. But that paper basically only looked at coding regions. They didn't look at the non-coding landscape. And so I think it'd be nice to revisit some of these other cases by looking at these putative regulatory regions for sure.
Yeah, that would be very interesting. I was also wondering if could trans-acting factors have contributed to the accelerated evolution of these conserved elements at all? Have you looked at that?
Yeah, that's a great question. I mean, that you probably appreciated that our enhancer screens are really not able to distinguish between change of the enhancer itself as a driving changes in gene expression versus changes of, for example, transcription factors or other things, transacting factors. And so, yeah, I think that's definitely a possibility. And of course, if the transacting factors change, then we might expect the element as well to change, sort of in a coevolutionary kind of thing. And so, but I think that that's a really good point, and it's something that we're hoping to do some more developmental work, and I think that would be a really cool possibility to look at that. In fact, there was a paper we did in collaboration with Cliff Tabin's group by one of his postdocs and Phil that found a transcription factor that had a lot of motifs in one of these elements, and that the results there imply that there could have been some change in the transacting factors as well. That was in Current Biology last year. Yeah.
Great, thank you. Do you believe that certain traits are more likely than others to evolve through regulatory changes compared to protein changes?
That's a really cool question. And I think that that's a major goal of comparative genomics in general and sort of mapping of traits. And, I think what we need to do is accumulate a bunch of examples. I mean, we know, for example, that coat color and things like that, plumage color to some extent, is a very genic phenomenon. And yet, if you look at the examples of coat color that are out there, it's pretty clear that you've got to have some kind of epigenetic or regulatory phenomena going on, because it's not like you get a completely melanic individual, which would imply that the gene is globally controlling, a gene like MC1R is globally controlling plumage or pelage color. And so my guess, honestly, is that regulatory elements are, on average, more important than coding regions. The taste receptor example, there's another example where you change the amino acid sequence, you change the trait. But a lot of it probably depends on how we define traits, whether we're talking about discrete traits or continuous traits. Certainly for continuous traits, I would imagine there would be a lot of regulatory evolution, but it'll be interesting to see what people find.
In relation to the PhyloG2P approach, someone is asking whether there is a requirement for a minimum number of species or phenotypes to be included in these kind of analyses to be able to get the most out of them.
I think, no, I don't think there's any requirement. I think the examples that we've seen are pretty good at showing that, well, with a small number of species, like, for example, there have been some nice studies in humans where humans are basically the only lineage showing a particular adaptation. And your power comes from looking across the entire genome where you have hundreds of thousands of potential events. Now, the approach taken by Gil Bejarano and his group, I think, has been amazing, because they're leveraging many species, such as mammals, simultaneously looking at many sites in the genome. And so I think you're going to have more power the more species you look at and the more whole genome across the genome sites you look at. But we've seen that I think you can make some progress with a modest number of species, such as has been done in human and primate studies, or, in our case, we only had 10 species. It was still a big study in terms of all the sequencing, but I think pretty soon we're going to have a lot more bird genomes, and this will really accelerate this whole PhyloG2P approach.
Thank you. I think we have time for one more question. How do we distinguish between genes that are important for a trait of interest versus genes that may be important for traits evolving on the same branches in a given tree?
That's a really good question. I mean, and for example, in our study, we found, some of these elements would accelerate on some rat-type branches, but not all rat-type branches. And we also, of course, we don't know what part of the anatomy or physiology a particular element is involved in. And that's where I think these epigenetic approaches can be useful, like ATAC-seq. ATAC-seq could tell us whether that element is in open chromatin in one part of the body, like limbs, forelimb, and closed chromatin in another part, such as the hind limb or other parts of the body. And that's, I think, where we can begin to narrow in on the spatial, where a particular element has a role spatially across the developing embryo. So I'm really excited to continue to use these approaches to refine this sort of PhyloG2P approach. It's not, I would say the PhyloG2P approach alone can get us so far, and we've got to eventually jump towards more targeted molecular approaches.
That's definitely very exciting. I think, unfortunately, that's all the time we have for today, but thank you very much, Scott, again, for accepting our invitation and for your excellent talk.
Thank you very much.
For everyone watching, do join us again on Wednesday next week when our speaker will be Dr. Daniel Matute from the University of North Carolina. Until then, have a great week, everyone. Stay safe and goodbye.